Nucleic acids are essential biological macromolecules that store and transmit genetic information in living organisms. The two main types are DNA and RNA. DNA serves as stable genetic storage, while RNA performs active roles within the cell. Nucleic acids are made of repeating units called nucleotides, linked into long polynucleotide chains. The nucleotide sequence carries the genetic instructions that control cellular structure and function. Now, before discussing their functions in detail, it's important to compare DNA and RNA directly. DNA and RNA are structurally similar, but they differ in several ways. DNA is usually double-stranded and forms the double helix structure, whereas RNA is generally single-stranded. DNA acts as the storage molecule for genetic information. Because it is chemically more stable, RNA is less stable but much more flexible functionally, allowing it to participate in different cellular processes. Another important difference is in the nitrogen bases. DNA contains adenine, thymine, guanine, and cytosine, while RNA contains uracil instead of thymine. In addition, the sugar in DNA is deoxyribose, whereas RNA contains ribose sugar. These differences affect both molecular stability and biological function. Now let's examine where DNA is actually located inside different types of cells. In eukaryotic cells, most DNA is enclosed within the nucleus, where it is protected and organized into chromosomes. However, DNA is not limited only to the nucleus. Mitochondria also contain their own circular DNA. And in plants, chloroplasts possess independent genetic material as well. This supports the endosymbiotic theory, which suggests these organelles evolved from ancient bacteria. In contrast, prokaryotic cells do not have a membrane-bound nucleus. Their DNA is located in a region called the nucleoid, where it exists freely within the cytoplasm. Despite these organizational differences, DNA in both prokaryotes and eukaryotes carries the genetic instructions required for cellular activity and reproduction. Inside the nucleus, DNA is not randomly arranged, so next we'll look at how it is packaged. DNA molecules are extremely long, so cells must package them efficiently to fit inside the nucleus. This packaging occurs through association with proteins called histones. DNA wraps around histone octamers to form structures known as nucleosomes, which are considered the fundamental units of chromatin. These nucleosomes further coil and fold into higher-order chromatin fibers. During cell division, chromatin becomes even more condensed to form visible chromosomes. DNA packaging is not only important for structural organization, but also for gene regulation. Regions of tightly packed chromatin are generally less active transcriptionally, while loosely packed regions are more accessible for gene expression. Now that we understand DNA organization, let's focus on its molecular structure. DNA consists of two polynucleotide strands arranged in an antiparallel orientation. This means one strand runs in the 5' to 3' direction, while the opposite strand runs 3' to 5'. The strands twist together to form the double helix structure first described by Watson and Crick. The two strands are held together through complementary base pairing. Adenine pairs specifically with thymine using 2 hydrogen bonds, while guanine pairs with cytosine using 3 hydrogen bonds. The double helix also contains major and minor grooves, which are important because regulatory proteins bind within these regions to interact with DNA sequences. This structure provides both stability and accessibility for cellular processes. The next step is understanding the individual units that build these nucleic acids. The fundamental building block of both DNA and RNA is the nucleotide. Each nucleotide contains 3 major components: a phosphate group, a pentose sugar, and a nitrogenous base. In DNA, the sugar is deoxyribose, while RNA contains ribose. The nitrogen base is attached to the 1' carbon of the sugar, and the phosphate group attaches to the 5' carbon. Nucleotides join together through phosphodiester bonds, forming long polynucleotide chains. The sequence of these nucleotides carries the genetic information of the organism. Even very small changes in nucleotide sequence can significantly affect cellular function and may lead to mutations or disease. Now let's look more closely at the nitrogen bases themselves. Nitrogenous bases are classified into 2 categories: purines and pyrimidines. Purines include adenine and guanine, and they contain a double ring structure. Pyrimidines include cytosine, thymine, and uracil, and they possess a single ring structure. DNA contains adenine, thymine, guanine, and cytosine, while RNA contains uracil instead of thymine. These bases are extremely important because their sequence forms the genetic code. Specific base pairing between purines and pyrimidines maintains the structural stability of DNA and ensures accurate replication of genetic information. One of the key structural differences between DNA and RNA involves the pentose sugar. DNA contains deoxyribose sugar, while RNA contains ribose sugar. The difference occurs at the 2' carbon position. Ribose contains a hydroxyl group, whereas deoxyribose contains only hydrogen. This small structural variation has major biological consequences. The absence of the hydroxyl group makes DNA chemically more stable, which is important for long-term genetic storage. RNA, because of its hydroxyl group, is more chemically reactive and structurally flexible. Now let's examine how nucleotides connect to form nucleic acid chains. Nucleotides are linked together through phosphodiester bonds. These bonds form between the phosphate group attached to the 5' carbon of one nucleotide and the hydroxyl group on the 3' carbon of the next nucleotide. This linkage creates the sugar-phosphate backbone of nucleic acids. Because of this arrangement, nucleic acid strands have directionality, described as 5' to 3' orientation. This directionality is extremely important in biological systems, because processes such as replication and RNA synthesis occur specifically in the 5' to 3' direction. Finally, let's briefly focus on RNA as a structurally flexible molecule. Unlike DNA, RNA is usually single-stranded. However, RNA molecules can fold into complex secondary and tertiary structures through internal base pairing. This structural flexibility allows RNA to perform many different functions inside the cell. Messenger RNA carries genetic instructions from DNA. Transfer RNA helps deliver amino acids during protein synthesis. Ribosomal RNA forms the structural and catalytic core of ribosomes. Additional regulatory RNA molecules also participate in controlling gene expression. Because of these diverse roles, RNA is considered both an informational and functional molecule. Now let's summarize the major concepts discussed throughout this presentation. In summary, nucleic acids are essential biological macromolecules responsible for storing and transmitting genetic information. DNA serves as the stable storage molecule, while RNA functions in multiple dynamic cellular processes. Both are composed of nucleotides containing a sugar, phosphate group, and nitrogenous base. DNA and RNA differ structurally in strand organization, sugar composition, and nitrogen bases. The organization of nucleic acids, from nucleotide structure to chromatin packaging, is critical for maintaining cellular function and genetic continuity across generations.